Hi guys, here with the second video of Chapter 14, the genetics unit. And we're going to extend a little bit into inheritance patterns that are a little bit more complex than what Mendel had initially predicted. Now, Mendel, I believe, probably got pretty lucky in not only what he decided that he was going to use to do his genetics experiments, but how clean his data turned out to be. And not only that, but the manner in which the traits were inherited in the pea plants was just super clean. Okay? So rarely, as that statement in front of you says, rarely does the relationship between genotype and phenotype work out as simply as what happened in his experiments. There are many, many, many exceptions to Mendel's rules. And so when we speak of something like I talked about the other day, simple Mendelian genetics, what we speak about is the simplest form of inheritance. And just something as different as putting two genes on the same chromosome can all of a sudden throw Mendel's experiment and his data out of whack. Okay? So many of these inheritable characters are not actually determined by one gene with two alleles at all. Sometimes it's a combination of many, many genes. Sometimes it's a combination of one gene acting on one chromosome and another being expressed on a totally different chromosome that acts in a totally different way. Sometimes it's none of that at all. Sometimes it's a function of the environment playing a role in it. So in this section here, we're going to get into talking about all that. Okay. Now, before we do, I want to express to you an understanding of. An understanding of what I mean by an organism's phenotype. A lot of time has been spent talking about phenotype and what the appearance of something is, and oftentimes that's the easiest one to understand. But before we get into a lot of these physical appearance exceptions to Mendel's rules, because almost all of these are exceptions to Mendel's rules shown by way of physical appearance, because those are the easiest to see, right? But when we talk about the phenotype of an organism, so much more is included. It's the internal anatomy, it's the physiology, it's the behavior, it's what your organs look like inside. It's how your body goes through its chemical reactions, the physiology. It's how you act. It's all of these things balled up into one are your phenotypes. They are all things that can be governed by genetics. For instance, my internal anatomy, you can't see that. Yes, it's a physical appearance thing. If you cut me open, you could see it. But I will always remember this one time, this random, weird time, I went to the doctor to get my lungs x-rayed for some reason, for bronchitis or whatever it was. And the x-ray technician said, "Wow, you have really long lungs." And I didn't have any clue what that meant, but she's seen her share of lungs, I'm guessing, right? And so she could tell the difference between my lungs and maybe somebody else's lungs. My lungs were longer than other people's, or some other people's. And so my phenotype for my lungs was different, which means my genotype might have been different for my lung type as well. So we have to extend this idea of phenotype more than just what we look like, because it is much more. It's how we behave. It's how our body works. It's everything. Okay? So keep that in mind, because we're not going to talk a lot about the genetic aspect of all of these things and how they manifest in the phenotype. But it is important that I think you understand that it is there. Okay? So without further ado, let's get into extending Mendelian genetics for a single gene, because we're going to talk about some with multiple genes as well. Inheritance of traits by a single gene may change from simple Mendelian patterns in the following situations. Sometimes alleles are not completely dominant. When we learned about Mendel's pea plants, he looked at traits where, for instance, the purple flower color was completely dominant over the white flower color. So there are times when that's not actually true. There are times when a gene has more than two alleles, okay? Blood types, for instance. When a gene produces multiple phenotypes, for instance, skin color. So we're going to touch on a lot of these things. We're not going to get super deep into a lot of them, but we are going to touch on a lot of them, okay? So first, let's talk about the spectrum of dominance. There we go. Complete dominance. This is an old baseball fight from my childhood, fond memories. of a 50-something, 40, mid-40s Nolan Ryan beating the crap out of 23-year-old Robin Ventura or whatever it was. Anyway, it was great. Anyway, what this illustrates is that one allele, oftentimes that codes for a specific trait, is going to be completely dominant over another allele, okay, which we call the recessive allele. And so this is the norm. This is how things normally work. But this is not always the case. This is where we get to these exceptions to some of these rules. Codominance is where we maybe have two genes or two alleles that are dominant equally, okay? Meaning that two dominant alleles will affect the phenotype in separate and distinguishable ways. So the flower that you see on here it probably is heterozygous for red and white color of petals, and certain parts of the flower are going to express the red, and certain parts of it are going to express the white, because no one knows who wants to win. They're kind of wishy-washy, like I don't know, maybe you can go and do it, maybe you can be dominant, and so they kind of both are dominant at the exact same time. And so you can see both of those traits represented in the organism at the same time, okay? Incomplete dominance is a little bit different. Incomplete dominance is where the phenotype of a dominant and recessive trait get mixed. So this one's even more wishy, like, okay, I kind of want to be dominant, and this one's like, oh, okay, you can kind of be dominant. And they're like, well, let's just work it out, and we'll be kind of dominant at the same time. And what happens is that those phenotypes, those traits actually get mixed. And so what you see here is if we had a red flower mating with a white flower, for instance, the heterozygous flower that results as the baby would look pink, okay? Because red color mixed with a white color, if you did it on a paintbrush or whatever, would look pink, right? And so there is some genetics behind this. If you look at the flow chart with the Punnett squares and the allele formations and things like that, or the gamete formations and things like that, illustrates what these plants would look like, right? So if we have the P generation where the white flowered plant is homozygous recessive for the W allele, for the white allele, and the red flowered plant is homozygous dominant for the red allele. Each one of those produces a gamete with an R or a W, and then they mingle. The resulting flower is going to have one red allele and one white allele, and that would create a pink individual. Okay, so we see this oftentimes, and a lot of times in plants actually, and it indicates that there's no real dominant allele, no completely dominant allele like we saw in the first example. Okay. On top of this, there's some more exceptions here. The frequency of the dominant alleles is not always the most common in a population. This is always kind of a paradox for me to think about. I'm like, oh well, we all have, for instance, 5 fingers. Okay, I got 5 fingers. Most of us have 5 fingers. Okay, I got 5 fingers on each hand. I got 5 toes on each foot. That's got to be dominant, right? Because it's normal, right? Everybody's got them for the most part. Everybody's got the 5 fingers, 5 toes. That's got to be the dominant trait. Well, it turns out that that's not always the case. In the instance of fingers, for instance, number of fingers, the number of fingers being 5 is actually a recessive trait. Okay, meaning the gene that codes for 6 fingers is actually going to override the gene that codes for 5 fingers. So if I had the gene that codes for 6 fingers, I would see 6 fingers before I would see 5 fingers. Kind of a crazy looking thing, but it's usually the case in a population where the dominant allele is the most common one, but not always. And so that's another one of those exceptions to Mendel's rules. All the traits that he happened to look at in those pea plants happened to be dominant, and they were the most common. Okay, it's not always the case. I will link a little reading. I think I've got a little reading that discusses the as to why people hypothesize as to why the 6-finger trait is dominant, but less frequent in a population. It's kind of an interesting read. So nothing you have to do, but I'm going to throw it in there just in case you wanted to extend your knowledge a little bit. Okay. So yeah, this is what somebody with 6 fingers look like. This guy is actually a pitcher all around the major leagues for a while. He actually pitched for the Cubs a little bit. And you can see this little guy here. There's one on his right hand, and I believe he's also got one on his left hand. So he's got 6 fingers, and that's not his pinky, but he'd have a little extra one right off the back end there. And it's most of the time that's what it looks like. Oftentimes these extra digits get removed at birth. The doctor will just take it off. The parents will say, "I don't want that thing on there," and then they'll just kind of remove it. Okay, it'll be a quick surgery. They'll remove it. Oftentimes, when you see something like this, it's not even functional. It's maybe a little flap of skin. There might be a fleck or two of bone in there. Maybe a little. You can kind of see a nail on one of his, but for the most part, they're not super functional. Just kind of flaps of skin with little bits and pieces in there. Unfortunately, so. Other times you get fully functional, and I think this is real. I think I got this picture from a reputable source. You look at his hands, and you're like, "Oh, why is he holding up his hands?" And then you take a second look at, "Oh, he's got a full-fledged extra finger there. It looks normal. It looks like he's got just 5 fingers, but then you look again, he's got legit 6 fingers. I bet he gives great back rubs." This one down here, possibly photoshopped. I put it in there for shock value, but. And I don't think there's anything this drastic where you get 1, 2, 3, 4, 5, 6, 7, 8 toes on a foot, but it could be photoshopped. I don't know, but I put it in there for shock value and entertainment. I guess I probably shouldn't have entertainment at other people's expense, but I throw it on there just so you can see. Okay, so that's what they call polydactyl, having that extra finger, and it is in fact a dominant trait to our recessive 5-fingered brethren. Okay. We also have traits that are governed by multiple alleles. Okay, so we just kind of talked briefly about some of the exceptions to the single allele or the double allele, single trait. Examples that go against what Mendel would think, and here we see example with blood types where we have multiple allele systems. And so with our blood types, we have the allele for A blood, we have the allele for B blood, and we also have the recessive allele for the O blood. Incidentally, AB blood is codominant. Okay, so somebody with AB blood will express both AB antigens in the blood. Okay, and we'll, if you want to know more about how blood typing works, we can do that. It's more of an immune system / human body system type of thing. But what you need to understand there is that the AB blood type does have dual expression. It has the A allele, which is dominant, and the B allele both expressed in the same individual. Okay, so we have multiple allele systems. Blood types. We've also got polygenic inheritance, which kind of work through multiple genes, not necessarily multiple alleles. So this is different. Multiple alleles. We've got 3 different versions of the blood type gene. Okay, we've got A, B, and then O. They're all blood type genes. They give you a certain blood type. But we also have this thing called polygenic inheritance, which is the additive effect of 2 or more genes, not necessarily 2 more alleles, but 2 or more genes that have a specific effect on a specific trait. In this case, the example they show here is skin color, and skin color works across a polygenic bell curve, and it is governed by how many versions of dominant alleles you get for at least 3 different genes. Okay, so the 3 different genes we see here are A, B, and C. And if we look at mom and dad, they're both heterozygous for this, and so they have kind of medium colored skin. And so they have that because there's a dominant 3 dominant alleles and 3 recessive alleles. The dominant versions of the allele give you darker skin. The recessive versions of the allele give you lighter skin. Okay, so effectively, the more dominant copies of a gene you get from your parents, the darker your skin is going to be. Okay, for these 3 specific genes. So if we look over here at the far right of the bell curve, we see that this individual would possibly have gotten 6 copies of the dominant gene, and that is very unlikely to get that based on this particular setup for these particular parents, which is why you see far less people with that super dark skin. Okay, on the far opposite end, we see that this full homozygous recessive for all 6 copies of the genes and the alleles is also very rare. It's very rare to get that combination of alleles based on these parents. And so it is also a very rare thing to see in the population. Where we see the more common scenario is that we get a heterozygous individual in the middle based on the heterozygous parents that were going to create it. So we see this bell curve for skin color. And so all sorts of things happen along these types of bell curves. I believe height in human beings can be similar to this as well. So those are some things to keep in mind, some exceptions to Mendel's rules. Okay. There's another phenomenon known as epistasis. Epistasis is where a gene at one locus alters the phenotypic expression of a gene at a second locus. Okay. For example, this does happen in mice with their coat color, and it depends on two specific genes. One gene codes for pigment color, okay, whether it's black or brown. Okay, that works on simple Mendelian genetics, right? It's a simple dominance. Big B is black is dominant over little b brown. But another gene located in the genome tells whether or not the pigment actually gets deposited onto the hair or into the hair. Okay, and so again, that works with simple Mendelian inheritance patterns, where if you get a dominant version C, big C, then the hair is actually going to get the color. If you get a recessive, okay, double recessive copies, then the hair is not going to get the color. Okay, so in class I'd be asking you some questions about this and things of that nature, but I want you to kind of just answer these, pause this on your own. Okay, if we look at this mouse here, so we got these parents. Okay, they're both black mice, and they both have color deposited in their hair. Okay, and we know this because the big B is dominant to the small b, so it's going to have black color, and it has a big C, which means that black color actually gets put into the hair. Okay, so I would ask you, okay, look at the genetics of this mouse. Okay, I would ask you, what color should that mouse be? You would tell me black, right? And then I'd say, should that mouse get the hair on its, or should that mouse get the color in its hair? And you'd look at the C's and you'd tell me yes. Okay. All right. Let's look at this mouse right here. Okay, this mouse right here is small b, small b. I would ask you, what color should this mouse be? And obviously, you know what color it should be because it's already colored. You'd say, it should be brown, Mr. Sutherland. Okay. Should the mouse get the color in the hair? And you'd look at the big C, it's big C, and you say, yes, Mr. Sutherland, the mouse should get the color in its hair. So the mouse is born with brown hair. So then we look at this mouse right here. This mouse right here, I'd ask you, hey, what color should this mouse be? And you'd tell me it should be black. And then I say, well, should it get that black color in its hair? And then you would look at the C's and say, no, Mr. Sutherland, it shouldn't, because it's homozygous recessive for the color deposition gene. So just because it gets a certain color, being black, doesn't mean that that black color gets deposited into the fur. Okay? Same way with this guy right here. This guy right here, I'd say, what color should he be? You would say, he should be brown. And then I say, is he going to be brown? And you would say, no, he's not going to be brown, because he has homozygous recessive for the color deposition gene, which means the color does not get deposited into his fur. Okay, so that's how epistasis works. It's a one gene affecting how another gene is phenotypically expressed. The gene of color deposition affecting whether or not the phenotype of color actually is seen in the mouse. Okay, kind of an interesting phenomenon. Now, I'd also like to talk here about the environmental impact on the phenotype. Okay, this is sometimes known as phenotypic plasticity. Okay, and so phenotypic plasticity can be seen in many different organisms, and one of the things that's really interesting to think about is how phenotypic plasticity affects human beings. Okay, phenotypic plasticity, or the environmental impact on the phenotype of an organism, oftentimes is referred to as nature versus nurture. This is kind of like the great experiment that scientists would love to run but can't really because it's unethical. They want to understand, are you a product more of your genes, or are you more a product of your environment, or is it a subtle mixture of both? And so one of the things that they do is they look at twin studies. And they also look at feral children studies. Okay, a couple of examples here. Here's a picture of a young lady from, I think, Ukraine, who was basically raised by dogs for like 8 years. 8 very influential, formative years in her development, from the time she was like 5 to the time she was like 12 or 13 or something, like 4 or 5 to 12 or 13. And so she had certain types of phenotypes that other people didn't. And some people think it was because of the environment. Did the environment have an effect on this phenotype? Did it even maybe have an effect on the genotype, which turns out can happen? And how would her phenotype differ from, this baby who's covered up by this black bar down here, with a nurturing, loving mother who would have taken care of the baby in different ways? How would they have differed, both phenotypically and maybe even genotypically? Twin studies are fascinating in this respect as well, because theoretically twins should have the exact same DNA, right? Or identical twins should. But even very early on, there's changes in their DNA and the way they're expressed. And oftentimes it's the environment's effect it's the environment that has an effect on this genotypic expression to produce the phenotype. I grew up down the street from identical twins. They were 2 of my best friends for a very long time, and I played with them a lot. I would go to their house before school, and we'd walk to school together. And my gosh, there was one who was just a crazy person, and there was one who I would consider relatively normal. And so what was it? What happened between the time they were born and the time when I used to hang out with them? Like, one of them would just go nuts, just insane. He'd want to fight people at the drop of a hat. He'd scream and yell at his mom and dad or whatever. And the other one was just kind of mild-mannered, and he's a really nice guy. Both nice guys to me, but was it the fact that the crazy one grew up on the west side of the house and saw too many sunsets, and that was depressing? And did the other one grow up on the east side and saw all these sunsets and was very uplifting and happy? Or sunrises and was very uplifting and happy? Who knows? Really don't. It could have been anything. But the idea is that there is some aspect where the environment does play into how your genes are expressed in some aspect, and also what your phenotype is under given circumstances. Okay, so some existing examples here. Hydrangeas, for instance. If you are a gardener, my wife loves to garden. We have hydrangeas in our garden, and the hydrangeas will change. Certain breeds of hydrangeas will change their color based on the pH of the soil. Okay, so sometimes if you have a pH of soil that's around basic to neutral, it's going to be a very, very bright pink like you see there. And other times, when the pH is acidic, it's going to be a bright blue. And then it's got this kind of gradation of color in the middle where it's a mixture of pink or blue. And it turns out that the pH of the soil plays a role in this because of the aluminum that might be in the soil. Okay? And it's this aluminum that gets taken up by the plant. If the pH is really, really low, it makes this aluminum more available. Okay, the acid makes the aluminum more available, and then the flower can take it up, and it's this aluminum that causes the color change to go to blue. Okay, so really it's not a genetic effect really at all when you think about it. It's a physical, environmental effect that changes the color of these plants. It's kind of an interesting phenomenon, but this is an example of this phenotypic plasticity in plants. We do see phenotypic plasticity in animals as well. I picked out just one example, which is the coat color in Arctic animals during different seasons. Here you see you see 4 different organisms here that all live in Arctic settings, and you would see different colors of coat during different seasons. So you'd see this bright white one. Obviously, we can kind of see them here during the winter times because everything's covered in snow. And you'd see these kind of mottled brown colored ones any other time, maybe fall or spring or sometimes even summer there. And this helps with camouflage. Now, I have not studied into how this happens or even why this happens, but I do know that it is the environment's effect on the phenotype of this animal. I've linked I don't know if it'll pop up, but I can put this in the the assignment notes. I've linked a paper that I got this graphic from that kind of goes into why and how this does happen when the seasons change, because these animals do shed their white fur for brown fur in these different seasons, and then get that white fur back when they are in wintertime. Okay? So that does it for video number 2. I will see you guys back here with video number 3.